In the first parts of this topic, we talked about the structure of membranes, movement of lipids within the membrane, and the movement of proteins in the membrane, lipids as well as proteins. Now we want to discuss about the transport of small proteins across membranes, which is also critical for the cell. So here is the outline of chapter 11 sections that we are going to cover, and this will be the last part of the module 1.2. So in the first section, we will have an overview of the transmembrane transport. So the cells can move substances across membrane by a number of processes. They can use passive diffusion, which does not depend on the existence of any protein. Then there are transport-mediated movements of substances across membranes. The characteristic of this transport is usually that the protein transporters are involved. There are three subclasses of those. There is facilitated diffusion, active transport, and coupled transport. And finally, there is bulk movement across the membrane, which is the movement of very large proteins and is usually achieved by vesicle budding and fusion, which is commonly known as the endocytosis and exocytosis, which is also very energy intensive, means requires a lot of energy expenditure to perform. So in this part of the discussion, we will cover the different ways in which substances can move across the membranes. So this is an overview of the membrane transport proteins, and cellular membranes are not only barriers but also conduits for selective transporting molecules and ions from one side of the membrane to the other. The transport proteins form a protein-lined pathway across the membrane that enables the hydrophilic substances to move through the membrane without contacting the hydrophobic interior. Three main classes of membrane proteins or membrane transport protein molecules are present, and ions and molecules actually cross the membrane using these membrane transport proteins. Each concentration of the substances also create of what is called a gradient across the membrane. The gradient across the membrane is indicated by these pink triangles with the tip pointing towards the lower concentration of the. It could be in the reverse orientation as well and depends on the substance that we are talking about. The first group of proteins that are called channels and facilitate movement of specific ions or water down the concentration gradient or down their electrochemical gradient. They can be non-gated, open all the times, or could be gated. That means they are only open in response to a specific chemical or electrical signal. And many times these are ATP gated. That means that they need a hydrolysis of ATP to keep them open. Otherwise, they will be closed. The second type of transporter protein are three types, and they also facilitate the movement of specific small molecules or ions. The unipoters are the ones which also transport just one type of molecule, or transport of single type of molecule down its concentration gradient. The other two types of transporter that you are seeing, these are called the co-transporter and known as either the symporters or the antiporters. The energy available from the movement of one or more ions down an electrochemical gradient drives the movement of one molecule against its concentration gradient, and this is also known as the secondary active transport. The third type is an ATP-powered pump, and the energy released by the ATP hydrolysis drives the movement of specific ions or molecules against their electrochemical or concentration gradient. This table actually shows the four mechanisms by which all the small molecules and ions are transported across the cellular membrane, and is pretty simple and self-explanatory. Just take a look at that. What are the different types of molecules that can be moved across the membranes? This portion actually shows the simple diffusion for the small molecules and the hydrophobic material that can simply diffuse, but we are going to consider these facilitated transport, active transport, and the co-transport this part of the course. So here we are seeing that some substance actually move readily and rapidly across the membrane by the passive diffusion. In this case, these substances are moving down their concentration gradient from high concentration to low concentration, so no energy is required. There are only a small group of substances that can cross this biological membrane, and these actually include the gases like a carbon dioxide, nitrogen, or oxygen. And there are some other small uncharged and polar molecules such as ethanol and to some extent water. And urea can cross the membrane from the region of high concentration to the low concentration, but this is very, very inefficient. So these substances move from a region of high concentration through the lipid bilayer to a region of low concentration on the other side of the lipid bilayer. The rate limiting step is the movement through the hydrophobic core of the biological membrane. So to summarize the rate of transport via passive diffusion, it is dependent on three variables. So the first is the concentration gradient. It is proportional to the molecule concentration gradient across the membrane. Second is the size of the molecule. Smaller the molecule are more permeable than the larger molecules. And the third and the most important is the molecule's hydrophobicity, and often called the rate limiting step of the passive diffusion. The hydrophobicity of the molecule is related to the molecule's partition coefficient, which is K. And again, repeating, the hydrophobicity of the molecule is dependent upon the K, which is known as the partition coefficient, which is the equilibrium constant of the partition between oil and water. In other words, the K, or the equilibrium constant, actually determines whether the substance is more soluble in oil or in water. A substance with higher partition coefficient is more soluble in lipid. Say vitamin A, which is a lipid-soluble vitamin. In contrast, say vitamin B complexes or vitamin C has a low partition coefficient and is more soluble in water. So vitamin A, being more lipid soluble, will pass through the membrane more readily compared to vitamin C, which is not soluble in lipid but soluble in water. So a number of substances that do not satisfy these three mentioned criteria, and passive diffusion is not sufficient for their transport. Large uncharged polar molecules such as water, glucose, many other sugars, vitamin like B complexes or C, many large ions and charged polar molecules such as amino acids, ATP, many sugars, phosphorylated sugars, say glucose 6-phosphate. These do not readily pass through the biological membrane by passive diffusion, but usually require the help of protein transporters. So we're going to take a look at these in a moment. So now let's talk about the facilitated diffusion, which requires the protein transporters and limited within a number of small molecules and hydrophobic lipid-soluble molecules. So certain amino acid and nucleosides, which are the building block of the nucleotides, and some sugars can be transported across the biological membrane by this process. In this case, the substance is moving from the area of high concentration to the area of low concentration. So it is still a diffusion and still no energy is required. The difference is that there is an assistance of some kind of protein transporter in the membrane. These transporters are usually referred to as unipoters because they can move one type of substance or one class of substance. So here are the differences between the facilitated diffusion and passive diffusion. First, the rate of movement for facilitated diffusion is faster than passive diffusion. And that is because the rate limiting step, which is the hydrophobicity, has been removed. Instead, the substance moving through the hydrophobic core of the lipid bilayer, the substance is actually moving through the transport complex, which is having an hydrophilic core. It is moving through the hydrophobic bilayer, but it is not in contact with the hydrophobic part of the membrane. It is actually exploiting the hydrophilic core of the protein to move through. As you have removed the rate limiting step, it is actually much faster. So the facilitated diffusion is also specific, which means that these unipoters only move one substance or one class of substance. And the facilitated diffusion occurs in a limited region of the cell where actually the unipoters or these proteins actually reside. So here is an example of a uniporter that is called GLUT1. We already talked about that in many different places. So the GLUT1 or glucose transporter 1 is a well-characterized transporter that moves glucose into the most mammalian cells by facilitated diffusion. There is really an interesting transporter. It can actually take on to two conformations. There is a conformation in which the glucose binding site is facing the outside of the cell. It is outward facing and shown here in 1 and 2, right here. And the other conformation, the glucose binding site, is facing inward or toward the cytosol as in 3 and 4, right here. Again, the glucose is moving from an area of high concentration to that area of low concentration, which is the cytosolic side of the membrane. This is how this transporter works. The glucose from the extracellular space will bind to the glucose binding site. When the glucose binds, this causes a conformational change in the transporter protein, and the glucose binding site is now facing inward. So the glucose is within the core of the transporter and it can diffuse into the cytosol. Once glucose is in the cytosol, it is often immediately converted to glucose 6-phosphate by an enzyme in the cytosol. This actually maintains the glucose gradient across the membrane, so the transporter maintains its functionality, or the GLUT1 maintains its functionality. Because what will happen that if glucose is moving from outside to inside, if the glucose concentration is actually increasing inside, then that transport will actually slow down. So it is immediately converted to glucose 6-phosphate. So glucose concentration is always high outside, and as the glucose coming inside the cell is converted to glucose 6-phosphate, the concentration is constantly maintained. So once the glucose is in the cytosol, the transporter can change conformation again, such that the glucose binding site is now in outward orientation once again. Now, if for any reason the gradient actually changes, or the glucose concentration is high in the cytosol and low in the extracellular space, this transporter will actually work in reverse. And in physiological condition, this actually happens, that the cells actually release glucose by this way as well. So here on the left-hand side is a structural model of the human GLUT1 uniporter in an inward open conformation towards the cytosolic phase. You can appreciate that this protein actually has 12 transmembrane alpha helical segments organized into amino terminal and carboxy terminal domains, N and C, right here, N and C, each of which consists of a pair of 3 transmembrane alpha helices. Those details are not required. So here you can see a cut open view of the structure, shows the surface electrostatic potential in yellow and blue. The hydrophobic amino acids are all in yellow, whereas the hydrophilic amino acids are in blue. So you can see that the hydrophobic amino acids, which are in yellow, facing towards the lipid bilayer, you can see that they are closer to the lipid bilayer, so they are interacting with the hydrophobic lipid core right here. Where you can see that the blue areas, which are the hydrophilic amino acids, they are facing either the exterior part of the cell, which are hydrophilic, or the interior cytosol, which is hydrophilic as well. And you can see the channel, this channel is actually lined by the hydrophilic or these blue residues, where the hydrophilic molecules can pass through this, which is a polar molecule like glucose. So to understand the functioning of the GLUT1, please recap from your prereq material about the enzyme kinetics, what is meant by the Km and Vmax of the enzyme. The Km is the concentration of a substance, here it is glucose, at which the rate of glucose uptake by this transporter here is half maximal or half the Vmax. And this depends upon the affinity of the transporter. So on the right-hand side, on the other hand, is a working model cycle of this GLUT1 transporter. We call this one as an alternating axis model, alternating axis from outside, axis from inside. So in the step 1, which is the outward open conformation, binds glucose, binding dependent upon the Km and the glucose concentration. And upon binding of the glucose, this transporter changes to conformation, as in step 2 of the ligand-bound occluded conformation. And again, it changes conformation to deliver the glucose into the cytoplasm. The release of the glucose is dependent upon the Km and glucose concentration as well, like in the step 1. The step 4 is the ligand-free occluded conformation. If the concentration of glucose is higher inside the cell than the outside, the cycle will actually work in reverse and will cause the net movement of glucose out of the cell. To prevent that, the cells actually already talked about, cells can phosphorylate the glucose to glucose 6-phosphate, which will not be transported by this GLUT family of transporter. So the human genome actually encodes at least 14 highly homologous GLUT proteins. These are named as GLUT 1, 2, 3, up to GLUT 14. So why we have so many different GLUT proteins? Each one of them has a different Km, which means that each one of them binds to the glucose with a different affinity, with high affinity, medium affinity, low affinity. For an example, GLUT2 in the liver has a Km of 20 millimolar. Increases the uptake after a meal when the blood glucose is about 10 millimolar, and releases glucose when blood glucose level drops to about 5 millimeter or below. The insulin actually regulates the insertion or removal of this GLUT4 into the muscle cell in the plasma membranes and internalized vesicle to regulate the glucose uptake. So these are some of the ways that GLUT numbers on the cell surface can be actually regulated. So we'll just come to this Km and the Vmax and the affinity and how these all correlate. So let's take a look at this. So here is what facilitated diffusion of glucose looks like compared to a passive diffusion of glucose across the biological membrane. On the y-axis is the initial rate of the glucose uptake right here in the y-axis. On the x-axis, you can see that this is the increasing concentration of glucose. You can see that passive diffusion is a very inefficient way for moving glucose across the plasma membrane. But if you can see that if GLUT1 or GLUT2 is incorporated, that increases the glucose transportation across the membrane many folds. And this is actually a very classic example to compare the passive diffusion with the facilitated rate of diffusion. So, of course, this kind of experiment would have been done with a liposome. So you actually manufacture this liposome artificially and put this GLUT1 and GLUT2 transporter in there, because you cannot control these experiment properly in a cell, in an isolated cell. So you do this in the liposome with these transporter proteins embedded into the liposome. And you can see that these are the passive molecules, or the passive diffusion will move some of these molecules across the membrane as well. And your question could be, okay, all right, you have already said that glucose is a polar molecule. How then even it is possible for moving the glucose by the passive diffusion through the plasma membrane? And this has to do with that sometimes with the energy given to the plasma membrane, you already have seen that these lipid actually just forms together, separate together. So if any kind of vigorous movement make a small hole transiently into the membrane, some glucose can actually move through. So this is only for the in vitro experiment, and rarely this will happen in reality on a plasma membrane. But again, remember that this experiment is in a test tube with the liposomes with these GLUT1 and GLUT2, and not in a physiological condition inside a real cell. So let's explain the figure a little bit more detail of the enzyme kinetics from your prereq material. The Vmax depends on the number of transporters. If you actually double the number of transporters, you will actually double the Vmax, or the maximum rate of glucose transportation. Here it is glucose. The Km right here is the concentration at which the transportation rate of a substance, here it is glucose, is half maximal, which depends upon the affinity of the transporter towards that molecule. Here it is glucose. So you can see here that for GLUT1, the Km is about, say, in between 1 and 2, about 1.5 millimolar, which means that GLUT1 binds to the glucose very efficiently, which is just 1.5 millimolar concentration. And it's a very high affinity. So GLUT1 actually have a very high affinity for glucose. On the other hand, if you look at this GLUT2, the Km actually falls outside this curve. So it's just about, this is still did not reach the half. So if we just extrapolate this line, which is half the maximal, maybe somewhere around 20, and the calculated value is actually for GLUT2, the Km is 20 millimolar, which is a very low affinity. So the GLUT1 uniporters are actually transport glucose into the most mammalian cells. And in normal physiological condition, the blood glucose is about 5 millimolar, and GLUT1 at 5 millimolar actually. So if you just go from here, 5 millimolar right here, it works at almost 70% of its maximal rate that GLUT1 actually works. But there are some other cases where the low Km is necessary for release of glucose into the bloodstream, and that's why they are important as well. So here, although water is not as large as glucose, but still it is a polar molecule and can pass through the plasma membrane by passive diffusion. So the cells have protein transporters for water known as aquaporin, found in all domains of life from bacteria to eukarya. The illustration shows the molecular structure of bacterial aquaporin. The one shown in the picture is actually a tetrameric protein having 4 identical subunits, and each of them has a hydrophilic water channel, and the water is actually shown with this blue dot right here. And the outside of this structure contain hydrophobic amino acids to interact with the phospholipid bilayer. We have seen several porine structure in the previous chapter. Please remember that the porine superfamily of proteins actually do have more than hundreds of different structures. And although we have seen a few of these beta barrel type of porins in the previous chapter, this aquaporin shown here is not a beta barrel structure. Instead, each of the subunits are composed of 6 transmembrane alpha helices and 2 shorter helices, which is buried half the way. These are the colored helices that you can see in this picture as the rod-shaped structure. So here is a beautiful experiment to show the role of aquaporin. The natural environment for the frog oocyte with which the experiment has been carried out is water. So to prevent the water from entering the oocytes, no aquaporin channel are present on the oocyte plasma membrane or the frog oocyte plasma membrane. But in an experiment, a frog oocyte was actually microinjected with aquaporin mRNA made in vitro, and the mRNA actually translates the aquaporin proteins inside the cell, and they will recruit the aquaporin on the plasma membrane, which is its natural destination. So the cells below are actually the control without the expression of aquaporin, and the upper is actually with the microinjected aquaporin mRNA. They are expressing the aquaporin inserted into the plasma membrane. The cells were placed from an isotonic, which is about 0.1 molar salt solution, to a hypotonic, which is a very dilute salt solution, which is much more dilute than the physiological concentration, about 0.035 molar salt concentration. So within a few minutes, you can see that the aquaporin transport water inside the cells, and the oocytes start swelling, and within a few minutes, the cell actually bursts. And this is actually known as osmotic lysis. So this actually gives you a classic example, the role of aquaporin transporters to transport water from outside to inside. Remember that we placed the cells in a hypotonic medium. That means the water concentration is outside the cell is much more than the water concentration inside the cell. That's why water will move from region of high to region of low, and that's through the aquaporin. So it still is a diffusion facilitated by the aquaporin protein transporter. So far, we have discussed the passive and facilitated diffusion, where the substance move down the concentration gradient and do not require any specific energy expenditure. Now we are going to talk about, and this is an uphill task. So in many instances, the cells need to move substances against a concentration gradient. This is carried out by active transport, and in this case, energy is required. In these examples, the energy is generated by hydrolyzing ATP. So active transport is carried out by what is known as pumps, which are ATP binding protein complexes that harness the energy in ATP hydrolysis to move substances against their concentration gradient. There are a number of classes of these pumps. There are P-class pumps, and these P-class pumps are responsible for moving a variety of ions against their concentration gradients across the biological membrane. they are usually made up of two identical catalytic alpha subunit and two beta subunit. But in this picture, we are showing just only one alpha subunit and a beta subunit though, but they are always in double. The alpha subunit actually contain the ATP binding and hydrolysis region, and they are really the workhorse of the pumps, which is in green. The thinking is that the function of the beta subunit is for the structural integrity to help maintain the interaction between the alpha subunit and the membrane. Another large class of pumps are the V and the F class of pumps. We're not going to consider in detail these pumps in this course, but just having a brief overview of their function. In general, both V and F class pumps are responsible for moving hydrogen ions or protons across the membrane against a concentration gradient. And they do that, and they do not have phosphoprotein intermediate, just like the P class pumps. So that's a very important difference. The V class pump actually couple ATP hydrolysis to transport protons against the concentration gradient, and they generate a low pH for plant vacuoles, lysosomes, and other acidic vesicle in animal cells by pumping protons from the cytosol into those organelle or the vesicles. So these are proton pumps to increase acidity into a certain part of the cell, particularly in an organelle or a vacuole. On the other hand, the F-class pumps works in a reverse way. It actually uses the energy in a proton concentration or voltage gradient to drive the ATP synthesis. So here the ATP is not used, but instead the gradient of the proton is actually utilized, so that due to that proton gradient, as the proton is moving down the concentration gradient, that will generate the ATP synthesis in mitochondria and in bacteria as well. So the third class of pump that we are going to consider and talk about detail as well is the largest class of pumps. Those are known as the ABC superfamily of active transport pumps. And ABC stands for ATP binding cassette. The structure of these pumps are very similar within this family. There are usually two large transmembrane region domains, which are labeled as T, and then two cytosolic ATP binding domains right here, known as A. But sometimes these A domains are also known as the NBD domains. NBD means nucleotide binding domain. Again, remember that ATP is nothing but a nucleotide. So these A domains, which are the ATP binding domains, are also known as NBD domain, NBD1 or NBD2, or A domain, A1 and A2 domains. Both of these conventions are followed, either A1, A2 domains or NBD1, NBD2 domains. And the transmembrane domains are called T1 and the T2 domains. So each ABC protein is specific for a single substrate or group of related substrates: ions, sugars, amino acid, phospholipids, cholesterol, peptides, polysaccharides, or proteins. So let's take a look at a couple of examples of these P class and the ABC superclass of pumps. So here, one pump that is well characterized and found in most animal cells is the sodium-potassium ATPase. It is a member of the P class pump family. What this pump does is that moves 2 potassium ions against the concentration gradient and 3 sodium ions against a concentration gradient simultaneously. Again, it harnesses the energy of the ATP hydrolysis, which is shown here. And you can see that this pump has 2 characteristic alpha subunit and 2 characteristic beta subunits. So let's take a look at the pump in a little bit more detail. So just like your GLUT1, these pumps take on 2 conformations. In this diagram, these are known as the E1 and the E2 conformation. So in the E1 conformation, there are 3 high affinity sodium binding sites facing the cytosol and 2 low affinity potassium binding sites facing the cytosol. So all 5 ion binding sites are facing the cytosol in the E1 conformation. Because these are high affinity sodium binding sites, the E1 conformation of this pump readily binds the 3 sodium ion. When this actually happens, the ATP hydrolysis event actually happens, and one of the aspartate right here is actually phosphorylated and on the transporter itself. So once this aspartate is actually phosphorylated, it actually changes conformation and it will go into an E2 conformation. In the E2 conformation, which is characterized by 3 low affinity sodium binding site on the outside of the cell and 2 high affinity potassium binding site on the outside of the cell. Because these sites have become low affinity sodium binding sites, the sodium will actually diffuse into extracellular space, and the 2 high affinity potassium binding sites readily capture 2 potassium ions from the extracellular space. And when this happens, there is a hydrolysis event that happens, and the aspartate becomes dephosphorylated, and you can see that the conformation will change back to its E1 conformation. So again, dephosphorylation of the aspartate changes the conformation to the E1 conformation. So at this point, what happens? The potassium binding sites become low affinity, and the potassium ions will diffuse into the cytosol. And again, the sodium binding sites will become high affinity to bind and repeat the cycle once again. This is a very active pump in most animal cells. Again, 3 sodium ions are pumped out of the cell and 2 potassium ions are taken inside the cell per cycle of the pump working. So this table actually shows a typical intracellular inside the cell and extracellular outside the cell ion concentration of several ions. So just for an idea, you do not have to remember those numbers, but I want you to remember the 2 key facts, which is true for every single cell in the metazoans. The ATP-driven pumps and the channels generate a huge gradient of ions between the cytosol and the surrounding extracellular fluid. Among those, the sodium and the potassium gradient is one of the most important, although the calcium chloride, everything is important, but these are the 2 most important ones, which maintain many biophysical properties of the cell, and including the cellular communication, excitability, and many others. The potassium concentration, just remember that the potassium concentration is much higher in the cytosol compared to the outside, which is about 20-40 times more than the extracellular fluid or the blood. In contrast, the sodium ion concentration is about 8 to 12 times lower in the cytosol than the extracellular fluid on the blood. So your take-home message, always remember that. We will come to this many times, so always keep this in mind, that the potassium ion concentration inside the cell is always high. Outside the cell, it is always low. On the other hand, the sodium concentration inside the cell is always less, and always outside it is more. So here is an example of the ABC superfamily pump. The ABC superfamily of membrane transport protein is the largest family of pumps, and it is found in both prokaryotes and eukaryotes. This particular pump was originally known as MDR1 pump, which stands for multi-drug resistant transport protein 1. The relevance of that will become clear in a minute. But today it is known as the ABC B1 pump. It was the first ABC pump that was identified. So what this pump does, it is able to take small charged substances as shown here and flip them in the biological membrane to the outer leaflet, or for inner leaflet to the outer leaflet, or the vice versa. And that's basically the function of the molecule. The ABCB pump is somewhat specific for a single substrate or a group of related substrate. It can move charged particles, ions, amino acid, phospholipid, or even proteins. It not only can move one charged molecule, but it can move a family of charged molecules as well. So this is quite relevant to chemotherapeutic intervention in patients that have cancer. Often the tumors actually become resistant to chemotherapy, and after chemotherapy is applied, that this resistance is actually facilitated by this ABCB1, and the name multi-drug resistance actually come from that. So this is how it works. Here is a cancer cell, suppose, and here's an ABCB pump indicated by this blue oval on the plasma membrane. Most chemotherapy agents are small hydrophobic or polar drugs or toxins, and these chemotherapeutic drugs can pass to the cell via passive diffusion. So if it is a hydrophobic, it can diffuse directly through the plasma membrane. And if it is hydrophilic, they need a transporter, and there are many different types of transporter that is being exploited so that they can transport these molecules inside the cell. So then they are pumped out by their concentration against their concentration gradient by the ABCB1. But what happens if the cancer cell is that there are spontaneous genetic mutation, that sometimes these spontaneous mutation lead to an increase of these ABCB1s. So right here. So you can easily see that a normal cancer cell without many of these ABCB pump is not being able to transport that many molecules. Whereas what happens that when it becomes a resistant cancer cell, a number of these ABCB1 increases, and very efficiently it will transport almost every chemotherapeutic agents that gets into the cell. So what happens that due to the mutations, these cancer cells produce more and more these ABCB pumps, and a significant number of these pumps actually pump out all these chemotherapeutic agents. So this cancer cell is actually showing that it's strongly overexpressing this ABCB1. The chemotherapeutic drugs enter passive diffusion, and then they are rapidly pumped out into the extracellular space. They are not able to kill the cancer cell because a lot of these agents are cell cycle toxin that block the cell division. When you talk about the cell division, we will talk about more about these chemotherapeutic agents. So you will notice that I put that resistance in the quotation marks. It is not that the cell are so much resistant, but the cell have such efficient pumping capabilities that because that they have so many of these pumps in the plasma membrane, that when a chemotherapeutic toxic molecules enter, it is rapidly pumped out. So for all intents and purposes, it is resistant, but really not really. So in cell culture, you can add enough of these chemotherapeutic agents to kill these cancer cells, to overwhelm the large number of ABCB1 pumps in the plasma membrane. But in vivo, you cannot do that, because then you would have to give a patient such a high doses of chemotherapeutic drugs that it will be lethal to normal cells as well, and that would be completely toxic. So you cannot. So for all intents and purposes, these cancer cells are labeled as resistant to chemotherapeutic agents or drugs. So let's see that how the ABCB binds to these wide variety of hydrophobic and hydrophilic drugs. So this illustration from your book shows the multi-drug transporter ABCB1, MDR1 as well, and with the ligand export. So the upper picture is actually a cut-out model of the ABCB1 protein, which is bound to two molecules of what is called as QZ59 SSS drug, which again, the name is not important, but this showing as a black structure in the pocket. So the important few points about this ABCB1 transporter is that this serine 289 and 290 actually affect the ligand specificity of the transporter, shown here in red. As we mentioned, it is a multi-drug transporter, and many different hydrophobic and hydrophilic molecules can bind in the pocket. So if the ligand is hydrophobic, it actually partitions in the inner membrane of the leaflet. It will partition in the inner leaflet, and it will move into the binding pocket of the ABCB1, where it interacts with the hydrophobic aromatic side chain of tyrosine and phenylalanine, which are in yellow portion of the cavity. Right here, you can see that. In contrast, if there's a hydrophilic ligand, it can actually directly bind from the cytosol. It doesn't need to be partitioned in the membrane. It can directly bind into the cytosol, and the binding of the drugs into the T domain actually triggers, which is a transmembrane domain, T domain, triggers the conformational change for ATP hydrolysis in the A domain. So the conformational change will be triggered by the ATP hydrolysis, and it provides the energy to change conformation of this ABCB1 or the MDR1, and it will expose the drug by flipping it towards the outside. And if the drug is actually hydrophobic, the drug will leave into the exoplasmic leaflet from this binding site to the exoplasmic leaflet, and then it will diffuse from the exocytoplasmic leaflet to the outside or the environment. So what happens is that if the drug is hydrophobic, it diffuses right outside from the membrane, and then on the other hand, if it is an hydrophilic, it will diffuse directly from this binding pocket to the exterior. So another ABC transporter is called an ABCB4, has a phospholipid flippase activity that we talked about in the last class. Here is an experiment which shows the flippase activity. It's a very clever experiment. This actually shows the ABCB4 in vitro and by a fluorescence quenching assay. So mammalian ABCB4 is expressed in a temperature-sensitive yeast mutant, and it's localized to intracellular endoplasmic reticulum vesicles in its normal orientation. And then the cytosolic surface of this vesicle facing outward is actually isolated, and you are actually carrying out this experiment in vitro in a test tube which is isolated from the yeast cells. At first, a synthetic phospholipid containing a fluorescently modified head group, the blue dots right here, is actually added into the media surrounding the purified vesicles, which actually will get incorporated into the membrane, because these membranes, these are hydrophobic, and this hydrophobic phospholipid get into the membrane. And you can see that you have a very high fluorescence when you have added these blue fluorescently labeled phospholipids to the membrane. In the step 2, what you do that there are two conditions. If you add actually ATP to the media, to the ABCB4, it hydrolyzes the ATP and it actually flips some of these labeled phospholipid into the inner leaflet. So into the inner leaflet. So they are now, these labeled phospholipids are facing the central lumen, which is the fluid field as well, central lumen. So from the outside, they flipped it towards the inner side. So on the other hand, what you see that if you don't add any ATP to the mixture or in vitro, the blue dots right here remain on the outside because there is no flippase activity of the ABCB4. So phospholipids containing the labeled fluorophores will not be flipped inside. They will remain outside. So this is the two differences in there. On the other hand, in the step 3, what you do, that a non-membrane permeating dithionate actually is added to the media, and the dithionate destroys the fluorescence of the fluorescent head groups by exposing it when it's exposed to the media. So this process is known as quenching of fluorescence. So dithionate is used as a quencher of fluorescence. So only the labeled phospholipid flipped into the inner leaflet will actually remain or fluorescing because the dithionate cannot pass through the membrane. On the other hand, all the fluorescence which is towards the outside or remain towards the outer membrane, they will be quenched by the dithionate or the quencher. So what you can see that due to the quenching, there will be a drop in fluorescence. But if you compare the liposomes which were added with the ATP and the ATP actually utilized by the ABCB4 to flip the phospholipids inside, the fluorescence is much higher than the samples where you didn't add any ATP, because there is no ATP means you have less fluorescence, which is right here, because all the fluorescent molecules still remained on the outside of the membrane and all of them are quenched by the dithionate. As they did not flip inside, they were not protected, so all the fluorescence from the surface is gone. On the other hand, in this one, why the fluorescence is at a higher level? Because all these fluorescent molecules are inside, flipped by the ABCB4 activity due to the ATP. And even after the molecules on the surface is quenched by the dithionate, there will be remaining fluorescence in there. That's why the level is actually higher. So what you can do that in the last step, step 4, can be used as a control, where the addition of the detergent to the vesicles generate these micelles and makes all the fluorescent lipid accessible to quenching agent, and all the fluorescence will actually be quenched and drop and merging them in the graph. So classic example of the flippase activity of the phospholipid flippase activity of the ABCB4 transporter. This table actually shows a selected human ABC proteins, and there are more than 50 different mammalian ABC proteins. The eukaryotic cell ABC proteins localize to the plasma membrane and intracellular organelle membranes, and they have a high expression in liver, intestine, and in kidneys, where the natural toxic and the waste products are removed from the body. You can actually appreciate from the list of molecules that this could be chloride, which could be really hydrophilic. Many are amphiphilic, and there are hydrophobic molecules. So ABCB4 does it all, transporting many different types of molecules and ions and substances. So in this section, we are going to talk about the transport or co-transport of the substances. So far, we have talked about moving substances across a membrane against a concentration gradient, harvesting the energy that is stored in ATP. There is another way in which the cells can move substance across the membrane against concentration gradients, and that is through the coupled transport. This is also very energy consuming because substances are moving against a concentration gradient, but in this case, the cells are exploiting the energy of a stored gradient rather than an energy stored in ATP. Think that someone pushes a revolving door for you and you piggyback and end up on the other side. And without pushing the door or spending the energy, you have reached the other side. And the coupled transport works the same way. So someone can push the revolving door and go the same way you are going. That will be going in the same direction. Or there could be another person coming from the other side pushing the door, and you are going in the opposite direction. And these are the two different types of transportation that we are going to see in the same direction. If two of them are moving in the same direction, we call it as a symport. If they're moving in different directions, we will call the antiporters. So in other words, when the substances move in the same direction of the transporter, as we said, that we call this symporter. And again, for the coupled transport, you need assistance from some kind of transporter. And when both of the substances move in the same direction, it will call a symport function, as we have seen. And in this example, the symporter move a sodium. So the symporter is moving a sodium in the same direction of the second substance right here against the concentration gradient, but in the same direction. So you can easily see. So what you are seeing, that the sodium is moving down the concentration gradient, and that energy of the sodium gradient is utilized by another substance moving against the concentration gradient. Here, the concentration of this black molecule or ion is less outside, more inside, and is exploiting the sodium gradient to move in the same direction. On the other hand, the cells can move substance in the opposite direction. So in this case, the sodium moving down the concentration gradient, and thus the other substance is moving from the cytosol to the extracellular space against its concentration gradient. See, the concentration gradient here for this black molecule or the ion is opposite from this one. So here in the cytosol, you have less concentration, and outside you have more concentration, and it's moving against its concentration gradient. So in this case, the transporter is known as an antiporter. So let's take a look at example of a symporter and one example of antiporter in detail. So one well-characterized symporter of intestinal cells is the sodium glucose symporter. This transporter can move the sodium down its concentration gradient and at the same time move the glucose against its concentration gradient. Now, what maintains the sodium gradient that is so critical to the purpose? So the sodium is typically high outside the cell and low inside the cell, as we have mentioned before. This gradient is maintained by the sodium-potassium P+ pump, as we discussed in the last part. So like the GLUT1 transporter, this transporter can also take on two conformations where the binding sites are either on the outside of the cell or on the inside of the cell. So in this first conformation, the binding sites are on the outside of the cell, and the transporter can bind two sodium ions and one glucose molecule, which is shown here. When this occurs, there is a conformational change of the transporter, such as the binding sites are facing inward and both sodium and glucose can diffuse into the cytosol. So the sodium moved down the concentration gradient and glucose moved against the concentration gradient. How was the energy in this sodium gradient harnessed? In all of these substances did not bind to the conformational change, would not have been taken place. So for example, if you lose the sodium gradient, it is very likely that this transporter will not work because there won't be sodium in the extracellular space to bind. So that is how the energy of the sodium gradient is harnessed to move the glucose against the concentration gradient. Two human sodium glucose symporters are SGLT1, found in the absorptive epithelial cells lining small intestine and kidney tubules, and there's another one called SGLT2 that is found in the kidney tubules. And their function is to resorb glucose into the blood from the forming urine. Now, inhibition of these symporters actually lead to excretion of glucose in the urine and the reduction of the blood glucose level. Now, SGLT2 is significantly more important, which is actually responsible for about 90 to 95% of the glucose resorption capacity, which is about 160 to 180 grams per day for an adult human being. So these symporters are the target for the treatment of type 2 diabetes, and several selective SGLT2 inhibitors. Actually, there are more than dozens are available in the market, and all of them are FDA approved prescription drugs. You can probably see that there is one called Invokana, and it's one of the most common commercial that you actually see in the TV all the time. Here is a molecular 3D structure of the 2 sodium Na+ and 1 leucine amino acid symporters from the bacterium Equifax aeolicus. It is composed of 12 transmembrane spanning helices. And so there are 3 membrane spanning alpha helices, and they actually bind to this sodium and the leucine. So you can see that right here, the sodium and the leucine. You can see that helix 1 is brown in color. Right here, you have the helix 6 actually is blue in color, and the helix 8 is in orange, and you can see right here. Whereas what you see that on the left, the helix 1 and 6 actually have a non-helical segment in the middle. So you have helix, no helix, then helix, helix, no helix in the helix. So you can see that there is a gap in between the continuation of the 2 helices for 1 and 6. So on the right, you can see that the helix 1 and 6 actually is coordinating a sodium, and right here is the leucine right here, which is making an interaction with the sodium with its carboxyl end. So this is how the leucine is actually co-transported with the sodium. And this is just to give you an illustration that how these are the different structure of the different symporters. You do not have to go into the detail of the structure, just gives the interaction of the 2 transporting molecules. So here is an example of an antiporter that is very well characterized and found in cardiac muscle cell. So here is a sodium-calcium antiporter, and in this case, the sodium is moved from the extracellular space into the cytosol down its concentration gradient, whereas simultaneously calcium is actually pumped out of the cell against its concentration gradient. So the concentration of calcium is more outside, less inside, and it's pumped out of its concentration gradient. Please remember that the P class pump that we talked about before moves the 3 sodium ions and 2 potassium ions in, moves 3 sodium ions out, put 2 potassium ions in. And this actually is responsible for maintaining the sodium-potassium gradient as well. So this entire system is actually exploited to treat individuals that are in congestive heart failure in an emergency situation. So calcium is really important for beating of the cardiac muscle cells, and you only need a relatively high intracellular calcium concentration in order for cardiac muscle cells to beat. When people have congestive heart failure, the calcium concentration is an issue. And so what happened that in an emergency situation, these individuals can given digoxin or oabin. So these are the 2 very well characterized pharmaceutical agents and they can actually inhibit the action of the sodium potassium P class pump. So when this happens, the sodium concentration actually increases in the cell, And when that happens, the sodium calcium antiporter becomes non-functional, because if there is too much sodium inside, there is no sodium gradient. Sodium cannot be going down the concentration gradient, and the antiporter will not work. The calcium concentration increases due to this in the cardiac muscle, and this actually facilitates the beating of these cardiac muscle cells. This helps the individual who are in congestive heart failure to help their heart cells pump again. So here is another very important use of antiporter, is in the carbon dioxide transport in the blood, which requires a chloride bicarbonate antiporter. So the gaseous carbon dioxide dissolves poorly in the aqueous solutions such as cytosol or the blood plasma, and change to highly soluble bicarbonate increase the plasma capacity to carry the carbon dioxide waste, which passively diffuse out of the cell. So approximately about 80% of the CO2 in blood is actually carried by the bicarbonate ions. So the systemic capillaries, which we are seeing right here, have a high CO2 and low oxygen in their environment. And this CO2 gas actually diffuse into the erythrocyte or the red blood cells across the plasma membrane. The enzyme, which is carbonic anhydrase, actually converts the CO2 into the soluble bicarbonate and H+, so it will be H+ and HCO3-, and H+ released in this reaction displaces the oxygen from the hemoglobin, and the O2 actually diffuses out of the RBC into the plasma and reaches the cells and delivers the oxygen. And an ion antiporter known as AE1 in the blue oval catalyzes the reversible exchange of chloride into the cell for the bicarbonate. Chloride gets into the cell, bicarbonate comes out of the cell. And this actually maintains a neutral pH in the red blood cells due to the chloride ion coming in. And the blood plasma dissolved bicarbonate right here circulates to the lungs. The lung capillaries, on the other hand, have a very low CO2 and a high atmospheric O2, just the condition which is just opposite to the systemic capillaries. And you will see a picture in your textbook, which is the red portion of the picture. So please take a look at that. So what happens in the lung capillaries, this low CO2 and the high atmospheric O2, just the condition different from this, the CO2 is excreted and the O2 is taken in. So the whole process is reversed, including the antiport. So here is also a nice summary that tells us how all these transport discussed so far maintains the concentration of ions and metabolites in the plant vacuole. There are several types of proton pumps which acidifies the vacuole right here. These are some of the unique ones that is only present in the plant, and these are the proton pumps as well. There are several protein channels like the chloride channels or the nitrate channels. These are all protein channels as well. There are proton antiporters, which move the H+ against the concentration gradient, which transports the sodium, that it can transport calcium. It can actually transport glucose by antiporting mechanism as well. So you can see that these sodium and the proton antiporters actually be overexpressed into the transgenic plants, which now can grow in high salinity or in high salt concentration near the coastline. So this is an overview of that, how many of these different types of protein transporters are responsible for maintaining an organelle and its condition. So in the next section, we are going to discuss about the transcellular transport with a few examples. We have seen in the plant system how many different transport systems come together in function of an organelle structure like a vacuole. And here we are going to talk about the transport across the cell in the mammalian system mainly. So I think you can appreciate that how of these transporters actually cooperate with one another. We saw that how the sodium gradient is so important for the sodium glucose symporter and the sodium calcium antiporter. The illustration here also shows the cooperation between several of these transporters. First, I want you to notice that these epithelial cells are actually polarized, and they actually do have distinct tight junctions and establish biochemically distinct apical and basolateral membrane regions that contain different transport proteins. This differentiation lipid the glucose transport through the paracellular pathway in between the cells. So that's the main reason that why they are differentiated. So here is a sodium potassium P class pump that maintains the sodium gradient across the cellular membrane. The sodium gradient is then exploited by the sodium glucose transporter to pump the glucose from outside to the inside of the cell. A GLUT2 transporter over here that is moving the glucose down its concentration gradient by passive diffusion. So here is the lumen of your intestine. Sodium and glucose symporter bringing the glucose from the intestine into the cell, and from the cell, the glucose is actually moving into the bloodstream, through the basolateral membrane, into the connective tissue used by the GLUT2 transporter. Now, this excess amount of sodium that is coming in for the symport is maintained by pumping this sodium again out by the sodium potassium pump, and the oncoming potassium is actually maintaining the proper condition of the cell. So what happens that now you can appreciate from here that the simple rehydration therapy depends upon the osmotic gradient created by the absorption of glucose and the sodium. So increased osmotic pressure created by the transcellular transport of the salt, glucose, and the amino acid across the intestinal epithelium draws water from the intestinal lumen, mainly through the tight junctions. And this is the reason for your sports drink to replenish this water. So here is another example, which acidification of the stomach lumen by the parietal cells in the gastric lining that happens after the food intake to kill the pathogens and to make the food acidic so that the stomach enzymes like a pepsin can function and requires this acidic pH. But the parietal cells acidify the stomach by hydrochloric acid while maintaining a neutral cytosolic pH. The apical region of the membrane actually contain H plus and K plus ATPase, or a proton potassium ATPase. So right here, you can see that the green one is the proton potassium ATPase. It's a P class pump, and this generates about a 1 million fold H plus gradient. On the other hand, what happened that the chloride channels right here actually maintains the pH. The chloride channel balances the H plus charge and preserves the electroneutrality. And here are some potassium channels right here, as well as to maintain the ion equilibrium. In the cytosol, the carbonic anhydrase, on the other hand, generates the bicarbonate from the carbon dioxide, and the excess of this hydroxyl is neutralized by in the basolateral membrane, the bicarbonate and the chloride antiporter actually transports the bicarbonate into the basolateral membrane, which is the way to transport the carbon dioxide through the plasma. So there is an additional example in your textbook which shows the functioning of the osteoclast cells, which also secretes the hydrochloric acid to dissolve the bone tissues for bone remodeling or say fracture healing, which sometimes even supply the dissolved calcium in case the blood calcium level drops at a dangerous level, which may lead to cardiac arrest. And also the calcium is essential for neural communication and as well as muscle functioning. So these are some of the transporters. Those are important for life of the cell and human health, because there are a number of genetic disease or inherited disease that are associated with the mutations in the gene that encode these pumps. And these tables on the next two slides really illustrate how important these pumps are. Let's take a look at a couple of examples. You do not have to memorize them, all of them. The ones in the yellow are actually the most important ones. For example, a large class of inherited cardiac arrhythmia that occurs due to the mutation in genes that encode potassium pumps, sodium pumps, or calcium pumps. There's another inherited disorder known as benign familial neonatal convulsions, which result from the non-functional potassium pumps. Myasthenia gravis cause actually severe muscle weakness, which you may have heard about. That is a mutation in a gene that encodes a sodium pump. The inherited disease that you may be most familiar with is cystic fibrosis. The pathology of cystic fibrosis results from a mutation in a gene known as CFTR or cystic fibrosis transmembrane conductance regulator that encodes a chloride channel, and that chloride channel is a member of the ABC superfamily of transporters. So let's take a look at how a mutation in this channel actually contributes to the cystic fibrosis. And here are some information about those. Some characteristic of cystic fibrosis: the primary cause of the disease is the production of actually a thick mucus that clogs lungs and many of the smaller ducts, which can lead to infection and pneumonia as well. There is obstruction of small bile ducts in the liver, the ducts of the pancreas, small intestine, vas deferens, and also malfunctioning sweat glands, all due to the production of mucus. Basically, all the small ducts and the glands in the body become clogged with a thick mucus. So here is the genetics of the cystic fibrosis. It affects mainly Caucasians. It is a recessive trait. 1 in 20 are carriers. 1 in 2,500 children are born with disease. And currently there are approximately 30,000 people living with cystic fibrosis in the United States. In the late 80s, there was a major effort to identify the gene that was responsible for the cystic fibrosis. So in 1989, it was identified as a gene on chromosome 7. The scientists were Francis Collins and Lapchi Siu. So Francis Collins was at the University of Michigan at that time when this discovery was made, and Dr. Lapchi Siu was at the University of Toronto, and they made a collaboration on this project. You may be familiar with Francis Collins. He is the current director of the National Institute of Health, big scientist, and he's also directed the Human Genome Project. So here is a structure of the CFTR, the chloride transporter. Again, it is a member of the ABC superfamily, so it has a characteristic transmembrane domains and the 2 ATP binding hydrolysis domains, and it is expressed in the lungs, liver, pancreas, intestine, reproductive tract, in the skin. So not surprisingly, you see the pathology of the cystic fibrosis. So you can see that the mutations that we usually see in the cystic fibrosis is the deletion of the phenylalanine 508th residue. And 67% of the cystic fibrosis is caused by this deletion. There's also another small percentage where a glycine 551 has been changed to the aspartate, and that mutation is also cause about 5% of the cystic fibrosis. So this illustration actually shows a simple cartoon of the functioning of the cystic fibrosis transmembrane regulator, CFTR. Please note that the ABC cystic fibrosis transmembrane regulator is a chloride channel, not a pump, means the transporter carries chloride down the concentration gradient and not up like a pump. But even with this down the concentration gradient channel, ATP or energy is required. The gating of the channel is done by the ATP, so you can call it a ligand, which is ATP gated channel, which is one of the very few ABC transporters which is a channel and not a pump. The CFTR need an additional regulatory domain in the cytosolic phase, which is right here, your A1 and A2, and this must be phosphorylated before the ATP is able to power the channel opening. So the first, what happens, that the A1 actually stimulates the binding of the A2 domain to the A1. First phosphorylation actually happens to A1, and that will actually stimulate that A1, A2 binding. And then the second ATP actually binds to A2, and then you have a tight binding in between A1 and A2. So when this tight binding actually happens, it actually opens the channel. As long as this tight binding of A1, A2 is in there, this chloride channel remains open. So if the hydrolysis of this ATP actually happens and these two are separated, the A1, A2 regulatory domains are separated, it will actually close the channel. So this is basically the functioning of the CFTR. So this is how the CFTR actually works in a normal lung and in a CF lung. So here are these are the endothelial cells that line the airway lungs, and these produce and secrete a mucus that ends up in the lumen of the airway. So what happens in a normal situation is that chloride channel is channeled into the lumen through the CFTR protein. Now that there is a net negative charge in this mucus that has been secreted by these cells, This then draws the sodium across this layer. And this sodium actually moves right here between the cells in the layer. And this type of transport is known as the paracellular transport. So sodium is drawn across to balance the negative charge. And so now you have a high sodium chloride concentration in this mucus. In response to the high concentration, water is drawn and water across water is drawn across this into the lumen in this area, and this actually hydrates the mucus, and it actually become much more lighter. But what happens in a CF lung, that there is a point mutation in the CFTR. We have actually talked about that. There is no movement of chloride into the lumen. So as the chloride cannot move into the lumen, there is no sodium paracellular transport sodium there as well. So there is no sodium chloride. As there is no sodium chloride, no water is attracted, and there is very thick layer of mucus which clogs these lumens, and that happen in smaller ducts as well. And this leads to a devastating physiological condition known as the cystic fibrosis. So the last form of transport that I just want to mention is shown in this one slide is bulk flow. The reason I'm just only mentioning it is because it's going to be the subject of the entire next topic. So the bulk flow is the movement of large fluid droplets or very large particles from the outside of the cell into the inside of the cell, or the movement of proteins from inside of the cell to the outside of the cell. These are commonly known as the endo and the exocytosis. So here is a couple of examples of bulk flow. When the cells internalize very large particles such as bacteria, the process is known as phagocytosis. When the cells actually move large fluid droplets across the membrane, this is known as a pinocytosis. Sometimes it is called as a fluid phase endocytosis, and that is an example of bulk flow. There are a number of cells that will secrete proteins, so exocytosis or secretion in this form is also known as bulk flow. And the proteins are moved between the organelles within the cell, which is also another example of bulk flow. All bulk flow is mediated by vesicle budding and fusion. So the vesicles will bud from one membrane, move to another membrane, fuse with that membrane, and release their content. This is true for phagocytosis, pinocytosis, where the vesicle bud from the plasma membrane and move into the cell. It is true for secretion, vesicles from inside the cell fuse with the plasma membrane, and is true for the intracellular transport, where the membranes from one organelle, say endoplasmic reticulum, fuse with another organelle, say Golgi or any other. So in this topic of this course, we will be focusing on the vesicle trafficking pathways. That will be pretty much the end of module 1.2.